So good afternoon, everyone. So I'm going to present you the strategic project INTERACTIF, which was founded in 2021 for 3 years, with the aim to study the interactive effect of deforestation and climate change on neotropical freshwater ecosystem, ecological networks, and ecosystem multifunctionality. So freshwater ecosystems such as rivers and lakes represent less than 1% of the total water of the planet and host 9% of all described species, that is much more species per unit volume than the oceans. However, freshwater are the most endangered ecosystem of the planet, and if you have a look on this graph, you can see since the 90s, the biodiversity of freshwater ecosystem has declined faster than the biodiversity of marine and terrestrial ecosystems. So freshwater ecosystems are mainly impacted by human activities at the local and regional scales, with water pollution, invasive species, flow modification, habitat degradation, and overexploitation. In addition, we can have also a global scale stressors such as warming, change in precipitation patterns, and also deposition of nutrients that can be superimposed to these local scale stressors. So freshwater ecosystems are impacted by multiple stressors, and this issue is particularly concerning in neotropical regions where habitat loss and deforestation will be very high, as you can see here in red and dark orange. And as well, it is expected a higher drought stress in the northeast part of the South America. So both multi-stressors will have a significant effect on the species extension on this area. So to date, most of the research was focused only on habitat or climate change, on biodiversity and ecosystem functioning. And only recently, ecologists were interested on the multiple aspects of climate change and habitat loss. And here we can see a study from Monticat and Pringle, who performed a meta-analysis with thousands of studies on different parts of the world on the effect of habitat loss on biodiversity. And they found that the effect of habitat loss on species density and diversity were greater in areas with higher mean temperatures and where mean precipitation has decreased over time. So this kind of biogeographical approach, however, does not enable to test the true pure and interactive effects of multiple stressors, because the study of real ecosystem is very complicated because a community are very complex and difficult to sample exhaustively and also to manipulate them. So here you have another example, who is a study by Pigot and collaborators, who study a stream-side mesocom experiment. So it's a mesocom experiment where the authors were able to manipulate different stressors, such as water temperature, deposition of fine sediments, and also, I cannot see, I don't remember what they use, and they changed the nutrient also excess. So in this study, they found that the warming interact negatively with nutrient excess and deposition of fine sediment on stream periphyton community, which is mainly bacteria, cyanobacteria, algae, and diatoms. So in this kind of approach, a mesocom experiment, so it's easy to manipulate and to test the different stressors on different communities. But in other side, with this kind of experiment, you cannot extrapolate the results on real ecosystems. So to study the effect of pure and interactive effect of different stressors on at the whole ecosystem level, in this study we focus on a natural microcosm with the use of tank bromeliads. So tank bromeliads that you can see here. So the leaves of this tank-forming bromeliad are organized in a rosette to collect water and detritus and to provide a habitat for aquatic microorganisms. You can have microorganisms where we can find bacteria, fungi, flagellate, ciliate, algae, and also macroinvertebrates. So this natural microcosm from the real ecosystem with co-evolved species, with simple communities that can be exhaustively sampled, easy to manipulate, with a high replication rate, and also it's also possible to measure different ecosystem functions. So in the interactive project, the main objective is to characterize the pure and interactive effect of deforestation and climate change on different levels of biological organizations in neotropical freshwater ecosystems. So this study is organized in 3 work packages, which in the thumb will provide a mechanistic understanding on how deforestation and drought will impact biodiversity, ecological networks, and ecosystem functioning. So in the work package 1, we'll study the response of community and ecosystem function to the different environmental stressors. In work package 2, we will infer ecological network for the different environmental stressors. And finally, in work package 3, we will study the response of ecological network properties and ecosystem multifunctionality to both experimental and predicted environmental changes. So for work package 1, where we are interested in the response of communities and ecosystem functions, we hypothesize that ecosystem will shift from detritus to algal-based food webs and from a dominance of macroinvertebrate to microbial assisted functions following deforestation. Also, we hypothesize that the food webs will disassemble from the top under drought stress because larger species will, which are larger energetic demand, and also these larger species have lower tolerance to water loss. Finally, we also hypothesize that the combined stressors will increase impact on biodiversity and ecosystem vulnerability, and we will test for the additive, synergistic, or antagonist effect of this combined effect of stressors. So for this study, we focus on the tank bromeliad Acmea aquilegas that can be found in both forested and open areas. And we perform an experiment where we can manipulate the deforestation. And here we select 50 tank bromeliad in the forest area. Here it's the bromeliad in green. And we select also 20 bromeliad in the open areas. And we start the experiment by the deforestation. And to emulate deforestation, we transplant the bromeliad from the forested to the open area. And after deforestation, we superimpose a drought trace in all the cases to end up with 7 treatments, as you can see here, and 10 replicate per treatment. So we have the pure effect of climate change either in forested and in the open area. You can have also the pure effect of deforestation and the combined effect of deforestation and climate change, either sequentially or simultaneously. So here it's an example of one bromeliad that was transplanted from the forest to the open area to simulate a deforestation. And at the beginning of the experiment in October 2021, we placed in each bromeliad a pre-weighted leaf litter inside of fine and coarse mesh enclosure to be able to dissociate the impact of macroinvertebrate and microorganisms in the leaf litter decomposition. Then in January 2022, we placed rain shelter above all tank bromeliads for the drought treatment. And in March 2022, at the end of the experiment, we collected all the leaf litter inside of the tank to be able to study the decomposition. And we also collected all the aquatic invertebrates by pipetting water inside of the tank. So all the invertebrates were sorted and identified at the lowest taxonomic level, and one specimen per morphospecies, and all the bulk were preserved in ethanol 96%. A part of the water was used to quantify the microbial biomass, and another part of the water was filtered to study environmental DNA. And in addition to leaf litter decomposition, we also measure the microbial respiration and photosynthesis and microbial carbon metabolic activities. Finally, in addition to leaf litter decomposition, we also add several additional leaf litter strips inside of both coarse and fine tubes to be able to quantify attached bacteria and fungi on the leaf surface, because they play a key role in leaf litter decomposition. And we focus on biomass and DNA analysis. So we still do not have all the data, but I will show you very preliminary results. So here, concerning the invertebrates, we can see that mainly the combined effect of deforestation and drought had a strong negative effect on abundance, on species richness, and on the channel diversity, but no effect on the Simpson. If we have a look on the invertebrate community structure, we can see that all the treatment has a significant effect on this structure. And as you can see here, for treatment 1, which is a bromeliad from the forested area, the structure of the community is very heterogeneous because of a high abundance here and a higher richness as compared to the other treatment. So if we remove this treatment, we can see here better all the other treatments and the great difference in the invertebrate community structure. So concerning the microbial community, here it's a PCR for the microbial biomass and abundance, and we can see an effect of all the different treatments, and mainly for the combined effect of deforestation and drought here in blue and purple. For the attached bacteria on the leaf surface, we also find a strong effect of the combined effect of deforestation and drought. Here with lower density of bacteria on the leaf surface, either in the coarse or fine mesh size, but we didn't find any clear pattern for the attached fungi on the leaves. Finally, for the ecosystem function, we also find a significant effect on the different treatments. And if we have a closer look on the decomposition, we can see here again that the combined effect has a strong impact and has reduced the leaf decomposition. Finally, concerning microbial carbon metabolic activities, we had a strong effect of treatment, and mainly for deforestation. This is the dot in light blue. So this result suggests that the microbial community has changed with the deforestation. And we will see now for work package 2, so the study of inference of ecological network for different environmental stressors. It's a work package led by Heidi Schimann and Lucie Ringer. So in this work package, we want to establish accurate macroinvertebrate and microorganism species lists and to assess the usefulness of water environmental DNA to characterize invertebrates and also to construct ecological networks. So in this work package, we hypothesize that higher invertebrate species diversity will be found based on environmental DNA metabarcoding because of the rare or cryptic species that may be missed in conventional surveys. We also expect temporal shifts in the communities after a change in the environmental changes. and as well as stress intensity increase from pure to interactive effect of the different stressor. Ecological network will be simplified because of a reduced number of species. So currently, we are at the DNA extraction stage for this project. So we have to do extraction for specimen, bulk, and water. And we want to build a DNA database for all the morphospecies we sample through conventional surveys to be able to refine the taxonomy of all these morphospecies. Then we will compare the list of species we will find with bulk and water environmental DNA to be able to assess usefulness of water extracellular DNA to characterize invertebrates, and also to be able to assess if we miss some morphospecies by our method of pipetting water and macroinvertebrates. So in addition to invertebrates, we will also focus on the prokaryotes and macroeukaryotes in the water to have and to be able to assess the whole community for all the microecosystems. So based on conventional surveys and on metabolic cooling approach, we will have co-occurrence data for multiple taxonomic, functional, and feeding groups. And based on expert knowledge, we will separate different feeding groups for invertebrate and microeukaryotes, and we will be able to separate predator from mesopredator and detritivores, which can be divided in filters, gathers, and scrapers, for instance. And for the microbial microeukaryote, we can have also micropredators such as rotifer and ciliate. We can have also detritivores such as flagellates, and also we'll base our study on different public databases such as Fungild and Bicrus to be able to assign different functional groups for bacteria, fungi, and algae. So this approach will enable us to construct true and interactive ecological network of reference. And in parallel, we will also use this metabolic cooling data to be able to build an ecological network based on different methods of inference. For example, with inverse covariance matrices, correlation approaches, or probabilistic methods. And we will compare all these methods with the network of reference to be able to assess the weakness and strengths of each of these methods of inference. And at final, we will keep the best ecological network that fit our expectation. And this final ecological network will be used for subsequent analysis in work package 3. So this work package 3 is led by Corinne Vacher and Olivier Deserald. And we want to characterize how ecological network properties and ecosystem function will respond to both manipulated and predicted environmental changes. So here we hypothesize that the pure and combined effect of deforestation and drought will lead to a substantial shift in ecological network properties. Ecological network structures will have a stronger indirect impact on ecosystem function compared to the direct impact of environmental stressors. And finally, we also hypothesize that the future climate change in the region, in the tropical region, will lead to higher cascading extinction that will propagate within ecological network and that's within the multifunctionality of the ecosystems. So here for each ecological networks, we will calculate different topological properties, such as the number of nodes or species, the number of links, its interaction between two species, either positive or negative, the connectance, which is all the possible links within the networks. And we also assess the nestedness and modularity, which give us an idea of the structure of the network, which can be either modular or nested. And finally, we will also calculate the multifunctionality, which is the aim of an ecosystem to assess to maintain different ecosystem functions simultaneously. So finally, on this work package, we also use different data on the previous study on drought stress and on result of this project to study both pure and interactive effect of different intensity of stressor on biodiversity, network properties, and ecosystem function and multifunctionality. And we will characterize the possible extinction, cascading extinction of the, and if they propagate differently or not within the ecological networks. And the difference in the propagation will be different according to the structure of the network, if it's nested network or modular, if the extinct species is a specialist or a generalist, and finally, if there is a functional redundancy in the ecological networks. So very quickly to sum up, so in this project, we have the ambition to study the pure and interactive effect of deforestation and drought on different level of biological organization. We want to reconstruct ecological network at the whole ecosystem level and to evaluate also the relevance of different methods of network inference. And we have the ambition to study the direction and magnitude of the variation in ecological network properties across different environmental conditions and to evaluate the possible cascading extinctions. And finally, we measured also different ecosystem functions that enable us to calculate ecosystem multifunctionality in relation to different ecosystem and ecological network properties. So I would like to thank all my colleagues that are involved in this project and you for your attention.
Our next speaker will be Tristan Lafont Rapnouil, who is also at AMAP, like earlier Céline Leroy, and he's doing his PhD thesis. And he will talk to us about the phenotype of a bromeliad species and whether it is influenced by maternal effects or the environment. The floor is yours.
Okay. So, yes, thank you for having me here. I can see that despite our love of science, there is more and more tired faces in this room. So we'll try to keep it as simple and clear for talking to you about some work I'm doing for my PhD, where we investigated environmental effects and the maternal effects on the phenotype of a bromeliad species. So maternal effects can be defined as the causal influences of the genotype and or the phenotype of the mothers on the phenotype of its offsprings. So basically, it can be seen as a form of transgenerational plasticity. And it is really important to understand them as they have major evolutionary consequences. For instance, they've been considered really important factors in local population and species adaptation, and especially rapid response to change in the environment, which is a crucial stake challenge in today's world. So in plants, they are even more important because as they are sessile, they cannot escape the environment. So they heavily rely on plasticity and plastic mechanisms to cope with the environment. And we can kind of distinguish three mechanisms of maternal effects. That would be epigenetics, that we have not looked in this study. You also have microbiota. So this is the microbiome that is transmitted from its mother to the offspring, and that can help and affect the offspring development. But we do not have yet the data on it. I just received the sequence. so you won't have any results about it today. And then you have the seed provisioning, so the mother can transmit to its offspring resources, nutrients, phytohormones that can affect the development and the health of the offspring and its success. So here we wanted to know how different abiotic factors could affect first the plant's phenotype, so the mother phenotype, the mother plant, the seeds it produces, so if different environment resulted in different seeds in terms of chemistry, and finally, of course, the resulting seedlings to see if there was kind of an effect of the past environment on the current success and phenotype of the plant. And to do so, we set up an experiment in French Guiana in Peru in greenhouses. So basically, we took seeds from one mother plant to limit the genetic variation, and we sowed them in different treatments. So 6 treatments in total, being 2 water levels, so 1 wet and 1 dry conditions, and additionally 3 different substrates. So a rich one being potting soil, a poor one being white sand, and an even mix of the both in yellow. And so these seeds were sowed, and we waited for them to germinate, grow, and then flower. So in Eichmer Mertensi, it's like about a year, which is very convenient to do transgenerational experiment and studies. So when the plants flowered, we measured a bunch of traits, ecophysiological traits, as well as chemistry, to characterize the response of our plant to the different environmental conditions. And here are the results. So don't be scared, I will walk you through this. So basically what we can see, it's a PCA on all the traits we measured. And what we can see is that we kind of have a nice discrimination along 2 different axes. On the horizontal axis, so on the left part and the right part, you have the water treatment effect. So plants growing on heavily watered environment and plants growing in dry environments. And we can see that they resulted in quite different groups, having more leaves, thicker, and with more water in it in the well-watered treatments, which makes sense. And then on the vertical axis, you have a gradient which is more representative of our different soils. So at the top, the rich one, which were heavily loaded in nitrogen, as well as phosphorus and chlorophyll. And at the basis, you have plants from the poor soils, which were more heavily loaded in soluble sugars, for instance, even though it's not really well represented on this axis. So we have an effect of our treatments on the plants. It was quite obvious when we entered the greenhouse. And then we wanted to see how it affected the seeds it produced. And here comes one problem. When you're working with life, it can die. So basically, what we had is that the plants from the dry treatment did flowered, but failed to produce viable fruits and seeds. So we only managed to have seeds for plants of the wet treatments. So from now on, you can forget all about water because we only have the effect of the nutrient availability in the soil. So we wanted to characterize the seeds to see if they have been provisioned in different ways according to the environment. And what we can see is, again, a PCA and a nice discrimination of our 3 groups, with seeds coming from plants on rich soil being richer in nitrogen, and plants from poor soils being richer in starch and poorer in nitrogen, for instance. Besides this chemistry, we also did a spectrophotometry analysis to check for differences in secondary metabolites that are useful for the seedlings development. So it was global, not very specific, but what we can see is that we have, again, very different profile of the seeds from rich treatment when we compare them to seeds from poorer treatments. So they have a really different composition in terms of secondary metabolites. And when we're going a bit more specific, we can look at some phytohormones. So here we have abscisic acid in its free and conjugated form, as well as salicylic acids that are important phytohormones for development. And we can see that the seeds have different composition. And now we wanted to see if it affected the seedlings. So for this, we crossed all the different environments to distinguish between the effect of the seedling environment from the one of its mother and of its origin. And then again, we measured a bunch of traits on leaf and the roots of 2-month-old seedlings. And again, don't be afraid, I will walk you through it. So we had 3 mother treatments, 3 seedling treatments, making 9 groups. So let's look at the right for now. We can see that on the horizontal axis, basically we have traits relating to biomass. So plants on the right would be bigger and plants on the left smaller. And on the vertical axis, we have traits related to the roots, and they won't be as important as the other in the following. Now let's look on the map on the individual projection. I figure that you can't see a lot, but we will start with the colors. So colors represent the seedling environments, so where it's growing. And we can see that seedlings in blue from poor environments are smaller than seedlings in red from rich environments, which makes sense, right? The more resource to grow on, the more growth. And we can see that yellow ones are in between. But what is interesting to see when we focus only on the blue, we can see that there is also an effect of the origin of the seedlings, so its mother. We can see that the ones coming from rich mother outperformed. They are higher biomass than the ones coming from poor mothers. This is also the case for the yellow ones, but we can see that this effect disappears in rich seedlings. They do not care if they were rich, if their mother were rich, if they are already rich, they don't care. So for a take-home message, basically in this study, briefly, the wealthier the mother, the healthier the offspring. So we didn't have like kind of specific effect where you grow better when you have the same environment as your mother. It's really about is your mother is rich or not. And these have some potential ecological implications, as bromeliads can be found in a very different substrate, different in richness. So having a rich, you can have a high diversity in the richness of the mother. I would like to thank INRAE for the fundings, our partners for chemical analysis, and of course my interns, which I work with for the greenhouse and field sampling. Thank you very much.
Thank you so much, Tristan, for keeping the time. The last speaker of today is Arthur Naas, who is at ISEM, and we are going to know something about the prehensile tail among Amazonian mammals.
So yeah. Hello, everyone. I'm Arthur Naas. I will present to you an overview of my PhD project, which is focused on convergent evolution of the prehensile tail among Amazonian mammals, and which is supervised by Pierre-Henri Fabre and Anthony Herrel and funded by the lab CEBA. So, okay. First off, a prehensile tail can be defined as a tail capable of grasping, and it is considered as an adaptation to an arboreal lifestyle. And as you can see here, prehensile mammals are fairly diverse, and most of those species are found in tropical forests. And looking at their distribution, most of them are actually found in the neotropics. And so, yeah, as I said, most of the species are found in the neotropics, and it's not only species richness. Looking at phylogeny of mammals here, you can also see that most of the independent apparition of these traits comprise neotropical species. And speaking of independent apparition, it quickly leads to the concept of convergent evolution. And in the case of my PhD, I mainly focus on morphological convergences that can be found between those taxa. And what is already known, I focus on two main structures, bones, more precisely tail vertebrae, and muscle, the tail musculature. And previous study I've already shown convergences regarding the tail vertebrae between prehensile carnivoran species and prehensile primates, and regarding tail muscles between the two independent apparition of the prehensile tail among New World primates. So speaking of muscle, it has only been tested in primates. So one part of the project will be to extend comparisons with two other groups, namely the myrmecophagids, or the anteaters, with two prehensile genus, Cyclopes and Tamandua, and one non-prehensile, the Myrmecophaga, and with a family of carnivorans, the procyonids, with the kinkajou here, Potos flavus, which has a prehensile tail, and two non-prehensile taxa, namely the coati and raccoon. And for this part, I will thank especially the Quata Association and the Institut Pasteur of Cayenne that allow me to access those specimens. So for this project, I won't be able to show results. I'm still gathering the specimens. Yeah. And more is known regarding osteological convergences, especially that those convergences are functionally linked with resistance to mechanical stresses. However, the taxa included in previous studies are often medium to large arboreal mammals, and small mammals have never been included in such analyses. And for this part, I will step aside a bit from the neotropics and focus on a tribe of rodents, mainly located in Papua and Australia, the Hydromini. And I will take this opportunity. As I mentioned, neotropics are the main hotspot for prehensile tails apparition, but it's more and more clear that Papua is a second hotspot. So if some of you have knowledge regarding meaningful comparison in canopy structures and especially connectivity between the neotropics and the tropical forest of Papua, I will be really interested to talk about this later. And so back to the Hydromini. So oftentimes when we want to do comparative analysis implying prehensile species, you found two main cases. Either you have in one clade one prehensile species or genus, or you will, like with the kinkajou, or you will find a whole clade composed of prehensile species like the didelphids. So the interesting part with the Hydromini is that here in green you can see the prehensile species, and you have quite a lot of apparition of the prehensile tail distributed along the phylogeny of the tribe, and they also have relative that are non-prehensile and arboreal, and that are non-prehensile and terrestrial, so it provides a good framework for comparative analysis. And lastly, as I said, those species are fairly diverse, and actually they might also be diverse in the way they use their prehensile tail. And if there is such a variation, we can ask ourselves how it can be associated with tail morphology, how tail use and tail morphology can be linked. And this question doesn't come from nowhere. It's on New World primates, for which we have the most behavioral data. What has been shown, for example, here, you've got Ateles, which is a species that rely really, really heavily on its prehensile tail, and comparatively, Lagotrix, which is a close relative, don't use it as often and as well, so to say. And when you look at their morphology, you can see that Ateles have bigger tail muscle, and that's even insert up to the hips compared to Lagotrix. And so, except for newer primates, we tend to lack behavioral data regarding tail use in postural and locomotory behaviors. And it will be the first step to link those two components of the phenotype of those species. And to do so, the idea is to extend our understanding of the behavioral diversity implying tail use in non-primate prehensile taxa. And to do so, we will use pictures from a camera trap placed in the canopy here in French Guiana that were kindly provided by the Ecotrop team from the Muséum National d'Histoire Naturelle. And yeah, for this, I especially thank Pierre-Michel Projet and Eric Gilbert, which have agreed to share these pictures. And this part of the project will take the form of an internship of 5 months, beginning in early 2023. So if there are master students interested in this project and working on those such data, feel free to come see me afterwards to discuss about it. Thank you for your attention.
That was a long day. Thank you very much for keeping it up. We are almost still 100 in this room. Quite amazing. We were up 120, 130 yesterday. So that's a great attendance, especially after 3 years without a presidential meeting. So it's really cool. Thank you very much for your questions, for your presentations. We're looking forward to next year's presentation and advances. Hopefully we'll get more of them. We also thank the different diversity of talks and thematics that we've covered over the past couple of days. It's really great. Yesterday evening, especially, we had this great small films, animated films. It was really, really cool to show how diverse the representation of biodiversity can be. So that's also a staple of CEBA, trying to not only look at one aspect of biodiversity, but all the different aspects, especially the representations of biodiversity by different cultures. And on that note, I'd like to conclude with an invitation to a show and visit the exhibit, which is just outside, called Kalalou. And if you have not done this, please do it. And so with this, I will close and thank you again very much, you all.